This method can help answer key questions in the honeybee nutrition field about the link between poor nutrition and hypopharyngeal gland growth and development. The main advantage of this technique is that one can use it to easily stain, visualize, and measure bee hypopharyngeal glands.
Generally, individuals new to this method will struggle because the hypopharyngeal glands can be difficult to locate and dissect without visual guidance.
Before beginning the procedure, pour melted cool-setting wax in a glass petri dish. Add 20 microliters of freshly prepared Giemsa stain into one well and 50 to 100 microliters of saline into an adjacent well of 1 microscope slide per nurse worker bee to be processed. When the wax is set, use 10 mm micro spring scissors to remove the head from the first bee, and use forceps, a dissecting microscope, and a wax carving pen to embed the head front side up in the wax dissection plate. Insert pins in the eyes and mouth to secure the head to the plate, and use a sharp breakable razor blade fixed into a pin vise to make a 2 to 3 mm incision between the eyes and mandibles on each side of the faceplate. Gently run the micro scissors under the faceplate and cut the antennal nerve that runs between the antenna and the brain. Using fine forceps, grab the faceplate by the mouth and pin the tissue face side down to the wax with fine point forceps and a pin. Next, pipette 20 microliters of PBS onto the open section of the head. The hypopharyngeal glands look like a string of pearls and are located on top of the brain if intact. Using superfine point forceps, gently